Morning, my name is Rebecca Lee. I'm the CEO of Vivli. Delighted to have all of you here today for our webinar, Unlocking the Power of Data Analysis in Vivli. After the presentation, we'll be sending a link to the recording and slides. So I'd like you to, if you have questions throughout, you can post your questions in the Q&A function and we'll respond to your questions at the end of each of the segments. So you can, of course, post your questions in the Q&A function. What I'd like to do next is introduce each one of our speakers. First off, I'll give a very short introduction to what is available in Vivli for about 5 minutes, and then we'll move on to Nicole Harmon. Nicole, if you want to give a wave. Nicole is the COO of CDISC, and we're really delighted to have CDISC co-sponsoring this with us today. We then have David McAllister, Professor of Epidemiology at the University of Glasgow. We're really lucky to have David here as well. And then Jack Shostak, Director of Biostats and Operations at Duke and a CDISC instructor with us as well. So for any one of you that have already done perhaps any analysis in Vivli, or perhaps haven't thought about how you might get started, this is a really good webinar for you. I think you'll get some of the basics on this, or if you've already done some analysis but perhaps want to enhance what you've done, this is also a good webinar for you. So some of the topics we'll cover is understanding the types of data sets and supporting documents you'll be receiving after approval, understanding CDISC, SDTM, and ADAM, and how these standards could potentially make your analysis more efficient, understanding reproducible, safe, open, and efficient analysis with some considerations for secondary analysis within the Vivli environment. And after both David's and Jack's talks, we'll have time for Q&A. So let me just kick you off with just some idea of the types of analysis that researchers such as Dr. McAllister have done in Vivli. Just take a few moments to tell you what is available and then move us through to Nicole in the CDISC presentation. So some requests by therapeutic area, about a little over a third of the proposals that come in are in the area of oncology. And since that's a very active area of requests for us, all of the approved requests actually are published on our website. So if you want to have a good idea of what some approved requests look like, you can always go to to our website. Another active area is active areas are in endocrine and cardiovascular disease, followed by neurology, rheumatology, gastro, and psychiatry. We have lots of others, probably 21 other therapeutic areas that are represented. There are some other methods papers that are really interesting as well. Really interesting ones in the area of AI and machine learning. So that really rounds out the types of proposals that are coming into us by therapeutic area. So how this actually works for requesters, let me just step through this very quickly. Someone would come and do a search on the platform for an area of interest. For example, let's just say it's breast cancer, and requests the individual participant level data that you're interested in. So each proposal is reviewed according to a contributor's publicly stated requirements. You can find them directly on the website. The data from the approved proposal is then accessed in a secure research environment, or at times they can be downloadable, and then analyzed using analytical tools that we provide, and they can be analyzed and combined together. And that will, David will go into much more detail about how that actually can be done. And then you can disseminate the research results and move those on. So I'll give you an idea of what some of those might look like. So how does that come about? So today we'll be looking a lot about focusing on this box, accessing data and using tools to combine and analyze these results. So that's really the space that we'll be focusing on today: how to access data and combine and analyze data. So what are the data packages? What does it mean when you receive a data package? So we ask our contributors to provide what we call this data package set. So that includes the final protocol, the data dictionary or define file. That includes detailed descriptions of each variable in the data set, the SAP, so statistical analysis plan. Pharmaceutical manufacturers provide a CSR, a clinical study report, and then the IPD data set itself. So the final anonymized individual participant level data. And then alongside it rides the anonymization report that outlines the anonymization approach for each specific data that's being shared. Optional pieces that often come with this data include the analytical code, the software that they use to run their pre-specified analysis, the AR-ready or analysis-ready IP data set, and the CRFs, the case report forms used to collect the data described in the protocol for each participant. So these are some of the things that will be described today, and I just want to run through kind of this data package that is provided. So what is provided in what we call the research environment? We provide all the software and tools that you can use. This includes, a number of R packages, Python packages. Some people like to use SAS, and then what we call in the premium environment, Jupyter Notebook. So everything you see here is available to you to use for your analysis. How does this roll up? So in the secure environment, you may be asking for data from any one of our 55 members, so pharmaceutical members, academics, or foundations members, and they actually are provisioned into the secure environment within which you can work. This is just a schematic that shows someone that's requested data from GSK, Johnson & Johnson, and two of the NIH platforms. The largest one is BioLink for cardiovascular data, and Import, and that has come into their environment, and they're actually sitting here and working. So what does the output look like? So I'm showing here some recent publications from the Vivli platform. So you can see, for example, Dr. McAllister has a publication, and that involves data contributors from AstraZeneca, Boehringer Ingelheim, GSK, Johnson & Johnson, Lilly, Roche, Sanofi, Takeda, and UCB. Dr. Ben Horn has also combined data from AbbVie, Biogen, Johnson & Johnson, Takeda, UCB, and come up with a nice publication in the Gastroenterology Journal. So these are just examples of what people are publishing on and getting publishing on in different journals. So we have many hundreds and hundreds of publications coming out. And, when I was running trials, it's different than primary publications. You're running a trial and publishing on it. This is secondary research, and they're actually doing quite robust research in this area. So these are the research environments available and the compute sizes. So this is what is available to all of you. So I'm going to move on to Nicole, who will be sharing her screen next and talking to you about CDISC. Thank you, Nicole.
Thanks, Rebecca. Okay, let's see if we can get started. Pushing all the buttons and getting everything moved over. All right, are we good to go?
Looks good.
I'm going to assume yes. Okay, great.
Yes.
So Nicole Harmon, I am with CDISC, and we are a nonprofit. We are celebrating 25 years in 2025. And many of you on the call today are with some of our member organizations and some of our academic partners. And so we'll say welcome to you and thank you for participating and being a member. And then also just wanted to provide today a quick overview of ways you can get involved with CDISC after this webinar today. So David and Jack are going to give you all the good meaty details. I'm just going to give you some ways that you can access free tools and access the standards after this webinar so that you have that available to you. So we are a global nonprofit. We have been designed as a consensus-based nonprofit that is developing standards for the last 25 years. We have a small team originally started in Austin, Texas, but now we're throughout the US, UK, and the world with contractors and trainers, and many, many, many volunteers on all of our standards teams. Our goal, we've been developing standards for quite a few years, but our goal is to really take those standards and ensure that the data flows directly end to end from protocol through analysis through submission. So as we think about what is needed in the standards as they stand today, we are really doing everything we can to enable and automate and connect those standards all the way down to ensure that there are no gaps in the data as concepts flow, terminology flows all the way through the data life cycle. And one of those other goals is just that we want to engage and adopt and grow the impact and the amount of users in our community. And so we want to really have that engagement with all of our community and grow that community, especially also in our academic areas. We were originally industry focused, but we've always had the goal of having that learning health system where we're all working together on research. Our standards have been required for submission to the FDA and Japan's PMDA since 2016. And various standard packages are required over the years. There are certain ones we have our foundational standards and then some therapeutic area standards. And so the requirements for regulatory submission have been around for about 10 years now and still have an active relationship with the regulatory agencies about how we can make their jobs easier. And that's a lot of what Jack has done is actually even training FDA on reviewing packages of data now that it's standardized, as well as just making sure that then we have this great relationship across industry, academia, and the regulatory. As I mentioned, we are a member-based organization. We have over 450 members globally from a variety of different backgrounds. So pharma, biotech, more technology vendors growing as everything is moving in that direction as well. And then growing our academic and patient foundation support as well. So we've worked with many patient and private foundations that fund research and really encouraging that those foundations encourage the use of standards for any research that they fund. So then later when the data is in something like Vivli, and Vivli's been a partner of ours since they launched, in the importance of why standards are so important to being able to look across legacy data sets. So would love to have you guys learn more about membership and how we can engage you in being a member of CDISC. And then additionally, we have events around the world and around the year. We're coming up on our European Interchange, which will be in Geneva in May. Would love to see you there. We will have a specific academic workshop that is a pre-event to the conference. You do not have to attend the conference to attend the academic workshop, so be sure to look into that if that's something that might interest you. And then through the summer, we have a lot of our Asia events. And then in the fall, we'll be in Nashville for some fun. And we also will have an academic conference to kick off that event in October. So always happy to answer any questions if anyone's interested in kind of digging in more. Each of those conferences also have CDISC education and training opportunities. And so, today you're kind of getting the high level, get you started, hopefully excite you about the exciting world of standards. But then later we also have a more deep dive approach that you can do either virtually, on demand, You can have, we can set up private training to come into your organization and train a team on standards. And we also have added in the most recent years more hands-on training and implementation of the standards a little bit further than what we used to have if you're more familiar with our previous training. One of the things I wanted to also provide to you is please visit our website. We have so many tools that we've developed on our website that are free and available to you to help kind of move your efforts along without as to get you oriented and active in understanding the standards, and then of course specific to your needs. And so we have an entire section called a knowledge base, which is just tools and resources and details about articles that are have a variety of focus areas that from academia to submission needs, and real world data and all the different ways that you can get involved. And then we also have electronic case report forms that are already standardized. And so if not in the for your needs today as far as legacy data standard or legacy data that you'll be using for research, but when you're starting a study, these are available to you and you can look through them and use them for initiating a study. And then I just want you to know that we have on the website, the standards are free in many different formats through our CDISC library. And so you can, depending on your needs, we wanted to offer that as an opportunity for you to learn more about ways that you can access and download the standards and learn more. And that is it. So thank you so much.
Great. David?
Okay, thank you very much. Right. Okay, hopefully everyone can see my slides. So I'm really pleased to be able to talk with you today. We've spent lots of time and we've gained lots as a group from using Vivli. So it's nice to get a chance to give a researcher's perspective or a research team's perspective. My name is David McAllister, so I'm a professor of clinical epidemiology and medical informatics and also an honorary consultant in public health medicine. And I'm based in Glasgow in the United Kingdom. So we thought it would be helpful to give you a little bit of a background of where we're actually coming from. So my own research started with routinely collected healthcare data, kind of administrative data and the likes. So that meant that we were used to the idea of reusing and repurposing data and to working within kind of safe havens. So we brought that with us when we came to Vivli and to secondary individual participant data analysis. But there are differences. So we've had a number of publications, some general medical, some specialty publications, and really we've got three kind of main Vivli projects that we've worked on. We had one with 120 trials across a range of conditions, kind of chronic obstructive pulmonary disease, psoriasis, and so on. We have had a project based mainly around type 2 diabetes trials, novel anti-diabetics, about 103 trials, 92,000 participants. And then our more recent repository combines both of those. And we're using both of those to look at a range of kind of clinical and also methodological questions. We've been interested in studying multimorbidity, where people have got multiple conditions, frailty, and I mean that in the clinical rather than statistical sense. And we've been really interested in kind of trial representativeness and applicability. And as part of that work, we've often brought routine healthcare data alongside the trial data in order to make comparisons and better understand representativeness. We've also done some work a bit more relevant to trial conduct. So we've looked at, how things like multimorbidity affect people feeling trial screening, and we've looked at the effect of different factors on people dropping out of trials, and finally we've looked at heterogeneity of treatment effects. And we've managed to get funding from a few different funders to do this work. Wellcome originally funded my own early work in in secondary analysis of the trial data. Medical Research Council in the UK, we've had project grants, and we've also had a clinician scientist PhD fellowship, and then more recently we've had 2 PhDs funded through the MRC Trial Methodology Research Partnership. And just recently, one of my colleagues, Dr. Jen Lees, who's on the call today, has got a welcome grant to look at cancer and renal disease, and she's put in a Vivli application to look at that. And I have a colleague whose steering committee I'm on in Manchester, who's doing some similar work around representativeness and immune-mediated inflammatory diseases. So that's where we're coming from. I'm obviously not going to read all these out, just to say we've had lots of input from lots of subject matter and methodological experts in the work that we've done. So this talk is, as you know, largely operational. It's about kind of getting the analysis done. But I think it is useful to just reflect on, as an academic, what do you bring that's different to what the sponsors themselves have? Because obviously they designed the trial, they're working within the organisation. So what we're doing, I think, is sometimes we're bringing a different perspective. We might have different kind of questions. And we also have a different methodological perspective. I think if you think like a secondary data analysis person, you might choose to use the data differently. For example, we use concomitant medications to define comorbidities. That's a very secondary analysis, routine data kind of approach. Things that also require scale. So if you need lots of data from lots of sponsors, I think that's a bit easier for an academic to do. And also it's useful to think about what needs other data sources. For example, we've used kind of data and model outputs from representative samples of individuals in Wales, data from 3 million people in Wales, and brought that in and compared that with trial participants. So these are the sorts of things that I think can be fruitful, in particular, if you're coming from a non-industry background. And different people with different interests will have different things that they're bringing, but that's been ours. So, on to the main part of the talk then. What are the principles of good data analysis using Vivli trial data? So the answer to that is the same thing that are the principles of good data analysis everywhere. You want something that's going to be reliable, reproducible, you want it to be open in the sense of you want things to be as reusable as possible and discoverable, and you want it to be efficient, both in terms of efficient for human beings doing the work, as well as efficient from a kind of computational point of view. So if the principles are the same, why are we here? Why do we need to have a workshop on it? Well, it's because there are special considerations within Vivli and other similar environments. So coming from a routine data background, we were used to kind of standard terminologies like the International Classification of Diseases, but the standard data formats are much commoner in the trials, I think, than they are in the routine data world. So we've learned a bit about the standard data structures, and we're going to learn some more about that from Jack. Standard terminologies are crucial, so understanding things like medical conditions, both in terms of medical history at baseline and also people experiencing adverse events, are all coded using the MEDRA scheme. Drugs, the de facto standard for that is the World Health Organization Anatomic Therapeutic Chemical Classification Scheme, so it's important to know that as well. So terminologies and formats, super important in this environment. It's really important to realize as well, though, that people will have done some similar analysis or related analysis to what you've done before. So when you produce something, you're going to want to know how that relates to published results, which is an important thing to consider. The documentation is huge. Rebecca told you some of the types of documents. So clinical study reports are vast. The statistical analysis plans are huge. The protocols are huge. That's great and provide you with lots of opportunities, but you do need to have a think about how you're going to handle all of that data, particularly if you're doing a large-scale meta-analysis of the kind that we've been doing. Particular challenges: it is a controlled environment. You cannot copy and paste. You can get things in and out, but it's a controlled access, both getting stuff in and getting stuff out, so that needs some thought and planning. The set of tools you have access to in Vivli is excellent, but it is not universal. So if you're using kind of internet-based tools and things like that, you need to think about alternatives. Anonymization is an issue. The data are not just pseudonymized, they're anonymized. So that means sometimes the data is degraded somewhat, like putting age into bands and things like that. So you need to factor that into your analysis plan. Computing resource. There is a cost associated with the computing resource after a kind of grace period inside Vivli. So that means that either you have to have funds in place to use that computing resource if you want to run it directly inside Vivli. You cannot expect to be able to use institutional resources like a high performance computing cluster in your institution if you're an academic. And there's also issues of time limited access. So thinking about that, planning when, and as soon as you turn on that environment, the clock starts ticking. So you have to think about how you're going to have an efficient workflow with the amount of time that you have. So speaking of workflow, so here's an overview of the general kind of workflow that we've adopted and that I think is a reasonable description. First of all, you have a kind of discovery phase where you're really finding out what data are there in these trials and what have you been provided with, because there will be some redaction. There's a harmonization stage which flows on from that, where you're attempting to make sure that the variable names and labeling and units and so on are all consistent across your different data sets. There's an analysis stage, which will obviously vary considerably from project to project, but there are, I think, some common principles. Checking is, of course, really important. And then, for people that are not used to working in safe haven environments, there's this export phase, which requires a bit of thought. Now, I've got arrows looping back from checking and so on through all these earlier stages, because of course this is not linear. You discover problems, you go back and change things. But once you've done the export, you want to minimize that. You can, if you've made a mistake with an export, you can go back, but it's something you want to try and avoid for kind of reasons of disclosiveness and also just reducing the amount of work for everyone involved. You want to try and make sure that you minimize going back once you've done your export. Thinking about that workflow, there are really two main ways that you might go about it. And everybody who's familiar with meta-analysis will be familiar with this. There's a one-stage meta-analysis where essentially you fit a model, usually a hierarchical model, to all of your trials and you export the results. The other approach is to do a two-stage meta-analysis where you have a stage one, where you fit a model to each trial, and then you can export the model outputs, and that means basically the coefficients and the variance-covariance matrix of the model, and then you meta-analyze those model outputs. And so those are the two main approaches. Now, we would generally favour two-stage, and this comes back to the principles that we put up earlier. It's probably a bit more reliable in that it's easier to compare your outputs to published results. I would argue that it's a bit more reproducible. If you take out, as I say, if you take out these model outputs and you can provide those to other researchers and they can check that they can reproduce your stage 2 model, it's more open. Again, if you've exported the coefficients and the model diagnostics and different summary statistics, if you release these when you publish your paper, then it's potentially reusable for yourself and other researchers. There's a bit more flexibility. So for example, if a reviewer says, well, actually, we don't think these trials belong in this analysis, you can take them out at that point, you can do different types of sensitivity analysis. It gives you a lot more flexibility as well with the use of different tools. You're able to access your university's high performance computing resources. So for example, in our most recent paper, the one that Rebecca was telling you about, some of the models took quite a long time to run. And so being able to run those across all the different cores within the university that were already paid for from our point of view was really helpful. And finally, if you are bringing in trials from other locations, maybe from academic sponsors, maybe for a trial that you're running, it's easier to add that if you're doing a two-stage meta-analysis than a one-stage. There are occasions where you will want to do a one-stage. For example, we published a paper once where we did some STAN models looking at correlations between comorbidities, and we ran all of that in the high performance environment within Vivli, and that was very successful. But generally, if you can, I would favour stage 2 for the reasons that I've outlined. Okay, so we're going to get on to going through each bit of the workflow shortly. But first of all, I just want to share some general tools and tips. Okay, so there is a special tool if you're dealing with trial data. And for those of you who are not familiar with it, the Clinical Trial Transformation Initiative provide a really fantastic resource to be able to get the data out of clinicaltrials.gov into machine readable formats that you can work with. It is a relational database called AACT, and what that does is it has an update every 24 hours of the content of clinicaltrials.gov. So here is their database schema. You can get that on their website and zoom in. Obviously, you'll not be able to see it properly here, but what you can see is that it's very rich. Not only do you get data about all of the kind of study characteristics, phase, numbers enrolled, when it's been registered, and so on, you get some really useful metadata for working with the Vivli trials. So for example, you've got different IDs, as well as the clinical trials NCT ID, you might have different sponsor specific IDs, and it has lookup tables for that. It'll tell you about the study design, gives you data, you can extract the eligibility criteria for the trial. That's all hugely helpful. You get lists of the primary and secondary outcomes. All of that's really helpful for trying to understand the data that you're faced with when you're working inside the environment. As well as that, for the majority of the trials which share their individual participant data within Vivli, they have also posted results inside clinicaltrials.gov, especially the more recent trials. So that means that you get things like age, sex, race, ethnicity at baseline. You get attrition, you know, how many people have dropped out from the trial by treatment arm. You get adverse event counts, often broken down into different system organ classes. And you get the actual treatment counts for, like, if there's a hard outcome, you get counts of events and treatment comparisons, kind of effect estimates. And all of that you can access from a stats package like R or Python. So it's a really, really useful tool. Other things that you do not have inside Vivli that you need to think about how you're going to work. So most people, at least hopefully, are working with a reproducible pipeline. And they'll often have version control as part of that. They might have productivity software. So you need to think, how am I going to recreate that within Vivli? It's very stressful to work without version control if you're used to version control. So for example, what we've done is set up our own simple version control, just using scripts within R that kind of reads in the scripts, running it from the command line, and then creates binary files from them. And what that does is it means that we can kind of go back and see changes that we've made to our analysis and so on. You also need a communication strategy. So you can't just copy and paste a bit of code or a bit of data from Vivli and email it to a colleague and say, you know, I know you've been working with this data, but I think I found a mistake. Can you check it for me, please? So you need to find ways of communicating both inside Vivli and outside Vivli. So inside we use a kind of an actions README file and an actions completed. So that lets us keep track and work together. And the other thing to say is that in terms of tools, you can still get lots of CRAN packages, as Rebecca mentioned. And one set of packages I wanted to call your attention to, because you might not be familiar with it, is the Pharmaverse packages. Now, these are in various stages of development, but the useful thing is that they have been developed with the CDISC standards and with the analysis of these trials in mind. So there's lots of like really interesting tools. I would really recommend the DiffDF one, which allows you to compare data sets. But it's also just worth browsing through it because it gives you an insight into the way that the drug companies analyse their own trials and what their priorities and processes are. So that's a really useful perspective as you're coming to analyse these trials as a secondary data user, particularly as an academic. Okay, briefly, other suggestions for good practice are, at some point you're going to want to take your code out or different types of results. So it helps, it makes it easier if the whole way through you're coding with intellectual property and privacy in mind. So that means things like not hard coding subject identifiers into your code, even if they're anonymized, kind of avoiding sort of individual level stuff that could end up with individuals being rediscovered. Similarly, some of the terminologies have got fairly restrictive licenses. So again, you want to be careful about what you include in your code. A related concept is, I think it's useful to separate your metadata from your code wherever that's possible. If you can abstract your metadata off into text files, then it makes that kind of disclosiveness and final export stage much easier. And of course, more kind of routine things like you're going to want to use naming conventions. It's useful to have a high-level README that gives an overview of all the different components of the analysis, because it rapidly gets quite complex, as well as kind of low-level READMEs for each kind of discrete analysis. So if something's like, clean baseline data, if you've got a little project for that, you'd have a README in there, but you'd also have a higher level README pointing to that. Okay, so back to the workflow. So first of all, talking about discovery. Advice for discovery: there's great internal data, that Rebecca outlined to you, but don't forget about the external data, particularly in the form of AACTs, I'll talk about in a second. I think you want to learn about the data standards. I found out about the data standards about a year into, I'm embarrassed to say, about a year into my first ever analysis, but back when I first started using IPD in another repository. So it's fabulous to know a bit about it now. It's well worth creating a data dictionary, and just remember that some of the data will be redacted, that's an inevitable part of discovery. So that means that even where data are consistent between different trials, there might be inconsistencies because of differences in the way that the data have been redacted. And that's to do with the way that the redaction works. Basically, the responsibility of the people sharing the data is to redact enough information so that it's no longer anonymous. They can use a variety of different strategies to do so, so those patterns of redaction, you need to discover them when you're finding out what kind of data you have. So first of all, just to remind you about the Clinical Trials Transformation Initiative, I think a really good thing to do is when you've got an idea about what trials you think that you want to get out of Vivli, get all the NCT IDs, they're all on the Vivli website, and then read the data in from clinicaltrials.gov. That allows you to create your own metadata, so you can kind of understand the eligibility, understand the outcomes, understand how many arms there are, all of that information I was showing you previously. And if you do that at the application stage, you can actually upload all that inside Vivli. So you've got your own little kind of metadata inside Vivli to help you try and understand the trials. You can, and we have also extracted data from papers, published papers, although that is a bit more challenging because obviously it's not in a machine readable format and it's not in standard formats, but sometimes you need that as well. Lookup files. So before, when I was talking about trying to abstract metadata from code, so you can actually abstract the metadata all the way out of the Vivli environment. So, for example, we published a paper a number of years ago, and here's an excerpt from it at the bottom left. We were interested in comorbidity in clinical trials versus the community. So along this flipped y-axis here is the proportion of people with a comorbidity, and along the x-axis is different comorbid diseases. The black dots are the community and the red dots are individual trials. So you can see the proportion of trial participants versus the proportion of people in the community that's got each of these comorbid diseases. Well, the way that we measured these comorbid diseases was using concomitant medications. So that meant that we were able to, outside of Vivli, create a lookup file using the terminology for drug names, and then and also subsequently for medical conditions. We were able to create this table and then just upload that into Vivli and join it on. So that very quickly showed us which trials reported a range of concomitant medications and which trials, for example, some of the cardiovascular ones, restricted themselves to cardiovascular and metabolic drugs. And the nice thing about that is it's self-documenting, and the work of data discovery quite quickly transforms into the work of data harmonization. Okay, now a really useful thing to do, so yeah, all the documentation is great, but often you have to open PDF files, and sometimes you have to open Word files to then click through to get into PDF files, and it can be very time consuming. So inside Vivli, we created a data dictionary. What we did is we looked through opening every data set and extracting metadata from those. And what I'm showing you here is a fake version of that I've created on my own computer to give you a sense of it. And I'm going to be brave and flip over to RStudio. So somebody shout if there's a problem. Okay. So here's our data dictionary. This is just for 2 trials, but inside Vivli we've got one for 231 trials. And what we've got here is we've got the path, so the original location of the data set, the file name. Most of the data that you're provided are in this SAS7 BDAP format. Now that's quite helpful because the variable names come in with variable labels and we're able to extract those. So we've got like variable name like study ID, domain, and variable labels that go with that. We've got the study ID, we've got the SDTM domain, which you'll hear a bit more about from Jack in a bit. We've got the number of rows in the table, the number of unique values. So here, for example, there's only one study ID, so that just appears once. The number of missing values, and then crucially, we also extract the first 1,000 individual values from any given variable. Now that's really useful because it means that you can quickly see which variables have been redacted. So you can kind of work interactively like this. So say, for example, you want to know where the age variables are. If I type in age, I can see that for both of these 2 studies in the demographics table, we've got age and it's measured in years. Variables like age are quite unusual in SDTM. Most often the data is in a long format. So for example, if you've got body system here. So here you've got body system here. The actual data is over inside the actual variable is in a long format. So say, for example, you wanted to find out, do these trials have measures of body temperature? So you can search inside the variables and then we can see, look, both of these trials, CDISC Pilot 01 and ABC, have both got body temperature. We can see that that's inside the vital signs table. So maybe we might want to have a look at those tables and say, well, we know it's got temperature. Does it have date? So we can have a look at date, and then we can say, good, we've got date and time measured, and we can see as well that it's not been redacted. You can also see here what's quite an interesting variation. So here we've got date for the CDISC Pilot 01, but for ABC we've got date time. So you can see that this is really useful for discovering data and continues to be useful as you progress through to harmonization. Okay, I'm going to flip back to the slides. Okay, as you work with these data, you're going to have to make a decision. Do I work with the raw data or do I work with the analysis ready data? Now the raw data tends to be more consistent across trials by design and within trials. So for example, the vital statistics table looks an awful lot, or sorry, vital status table looks an awful lot like the laboratory table, which looks an awful lot like the concomitant medication, and so on. They're all in these long formats and with similar names, so it's more consistent. It's also a bit more discoverable, and so for those reasons, we use those most often in our meta-analysis, where we're analysing lots of trials, and consistency is really important to us. However, not always. Sometimes variables are complex, particularly if you've got outcomes that have been adjudicated, or you've got variables composed from lots of different questionnaire elements. In those kind of scenarios, we went for the analysis ready. But that's a decision that you're going to have to make on a kind of study-to-study basis. And it might also depend on redaction. So sometimes the raw data might be redacted, but you've got the processed variable in the analysis ready, so you're still able to use that. Okay, so that's discovery. That kind of slides neatly into harmonization. Again, lookup tables are useful. So here's a fake example, but it's very realistic. We had a recent work on HbA1c, glycosylated hemoglobin. The names were different across the different trials. So this table, it has the trial ID, it has the original variable level, original value, and then we've got what we want to assign it to. And then all we need to do is join one to the other, and that allows us to relabel it. What's the point in doing that rather than just hard coding it? Well, it's language agnostic, so if you've got some members of your team working in R and others in Python and maybe some in something else, you've got that data. But it's also very readable for a human being. They can just open up the spreadsheet and see what it is without having to dig through your code. For the same reason, it's easy to export because the person at Vivli reviewing your data to check it's not disclosive is able to read it. And it's also self-documenting. Another kind of lookup that's pretty valuable, not for export, but for working within Vivli, is to have a lookup between the name that studies are given in terms of their folder names, their NCT ID, the one from clinicaltrials.gov, the various sponsor study IDs, and sometimes there's more than one. It's really useful having a lookup table between those. And sometimes as well, different participant IDs. So there are different ways that IDs are recorded, particularly with some of the way that some trials do the anonymization, there might be even more than that. So it is useful to have a lookup between different participant IDs as you're harmonizing data. Particularly if one member of your team, for example, might be harmonizing baseline data and another member is harmonizing outcomes, it's really useful to make sure that you know that you can join those together straightforwardly. Okay, so on to analysis. So this is more project specific. However, there are a couple of points to make. One point is you can reduce the size of your data when you're developing code. So take a random sample within arms of some of the participants in the trials when you're running it. Again, you're going to have to deal with redaction. So in an analysis we're doing at the moment, we are interested in duration of diabetes to see whether or not treatment efficacy differs by duration of diabetes. Now, most of the trials have given us diabetes to the nearest duration of diabetes to the nearest year, but some of them, for a particular sponsor, have binned that. Sometimes in 5-year bins, sometimes in 10-year bins, sometimes longer periods. So you have to come up with a way of dealing with that, of imputing it, but without attempting to re-identify. So that needs a bit of thought. Standardizing variables, subtracting the mean and dividing it by the standard deviation. It's a useful thing to do in regression modeling anyway, but it's particularly useful here because when you're thinking about exporting your data, getting the coefficients onto roughly the same scale is great because then you can export things with a sensible number of decimal places, which is consistent across different variables. But, and I'll just mention model outputs first. Getting model outputs out is relatively easy. There's a really nice package in R called Broom, which can help with some of this. You can get out the coefficients, you can get out the variance covariance matrix, although actually we prefer to turn that into a correlation matrix first and export that. You can get out model diagnostics. Nobody's too anxious about model outputs. It's pretty low risk that someone can be re-identified from model outputs from your kind of standard regression models or kind of even sort of longitudinal ones. What's actually much more worrying is summary statistics. And sometimes you have to be a bit creative as you're thinking about exporting summary statistics, and particularly when you're wanting to export diagnostics. Because if you think about it, quite often with diagnostics, you are comparing the data to your model fit. So there are ways that people could recover the individual data. So if you look over here on the right, here's an example that we've done recently. We wanted to get out an approximation to the empirical cumulative distribution function for age for each trial by sex. And these plots here, each of the blue lines is the empirical cumulative distribution function for age, and the red line is our approximation. And you can see in the top left here that the blue line goes up in bumps, and that's because it was age in bands. So we wanted to reassure ourselves and the reviewers that our approximation to the true empirical cumulative distribution function was reasonable. So the way that we did that was to plot it, but we took a number of steps to prevent re-identification. First of all, we removed the y-axis ticks and labels. We did the same for the x-axis. We stretched out both so that you couldn't say which trials had, you couldn't work out which trial it was because it had a narrower age range in its inclusion criteria. We randomised the order of the trials so that you couldn't just guess that, you know, from the trial ID numbers which one it was. And finally, we reduced the resolution in the TIFF so that you couldn't get to too fine a detail. So that might be in a bit of an overkill for this case, but it does illustrate that you do need to be a bit creative and thoughtful when you're trying to avoid disclosiveness. Okay, so checking. It's not our target that checking's important. We obviously want to check our alignment with published results. So I think it is a useful exercise to show that you can approximately replicate the trial main effects and main counts before going on to do the particular analysis you want to do, just to check for kind of coding and other errors. The reason I've said alignment rather than strict replication is because the anonymization might mean that you can't ever completely replicate the results. So how do you do that? Well, again, I would come back to using that AACT resource. If you've pulled in that metadata, then you can compare your results with the published results. You can also, if you get funny things in your data, like why are there no smokers in this trial, you're able to look at the eligibility criteria and say, well, that's because they've been excluded, and so on. Similarly, if you find that a trial is very limited in terms of the representation of people from different race ethnicities, you can have a look at the countries and see if that's the explanation why. Similarly, looking at titles and descriptions as well as some more detailed descriptions. One of the nice things as well is you might want to consider publishing some of these comparisons. So for example, my PhD student, Ryan McChrystal, who is working on trial attrition, the first thing he did was look at his numbers for people being excluded, people dropping out of the trial, and compare them to the published ones. And he's got a nice table, and when he publishes his paper, he's going to be able to put that in the appendix and reassure everybody that, that his data analysis is robust. Okay, and on to export. So, as I mentioned before, you want to be thinking about export from the very beginning. You've got to remember, I'm going to be taking these data out, so that's where your export conversation begins, and we've talked a bit about that. However, there are other little practical things to consider, like you probably want to have an export folder or a group of export folders within your Vivli environment, keeping it separate from your main analysis. When you go to do your export, you don't want to be opening up a folder and saying, oh, I wonder which of these files is safe to export, and you accidentally put in the individual participant data. When you're doing it, and what I would suggest as well is a good approach is if you have your kind of analysis projects, and then if you kind of programmatically write into your export folder, then that's quite a nice way to work. And then your exports themselves, you want them to be machine readable, of course, because you can't do your two-stage meta-analysis very easily if they're not machine readable, but you also want it human readable because a human being is going to have to open up your data and look at it before they press OK and let that come out of the environment and go to you. So part of that means including metadata, partly for the person reviewing it, but also for yourself. You know, often you'll have multiple exports, so you want to really know what is it I exported this time. You want a nice overview about the purpose and the content. You want a description of each kind of thing that you're exporting, whether it's an image, a figure, or it's kind of some summary data, and really a kind of row by row and column by column explanation of what you've got. Now, Vivli is very generous in its file export size, but just a note about some other repositories actually have quite strict limits on how big the files are allowed to be. So you might want to have a think about that, about the number of decimal places and whether you want to perform standardization to reduce the size of your exports. I think as well, you want to run checks on your planned exports. So if you've got your exports in a separate area in Vivli, you can then kind of do a mock-up of the analysis that you're planning to do, because you don't want to get the export out and then find out that you don't have the data you need. So I think it's very useful to, if you can, run a full version of stage 2 of your meta-analysis, or if not, see if you can run some sort of approximation to it. Like if you're running a Bayesian model outside, maybe you could do a frequentist one inside, just to check that you've got what you think you've got. And I think it's also useful just to open up those exports in the export folder and just check, is this what I think I mean to export, so that you can minimize that going backstage. If you have the resources, it's useful to consider independent review. So for example, that's what we like to do. If one person produces an export in our team, we like to have someone else look at it before we actually make the export request. And then when you do get your exports out, it's a good idea to place it under version control. Again, just so that you can keep track about what you got and when and how it's changed over time. Okay, and then one last thing, it goes a bit beyond the data thing, but it is a kind of data sciencey, informaticsy point. You have to share your planned public outputs with sponsors. They are entitled to review it and give you comments. So that means the paper and the appendices, that's straightforward enough. But really, we ought to be sharing our kind of code and data on GitHub or similar. But if you're going to do that, then you need to also create the same content for the sponsors to review. So the way we did that is, so for example, if we've got a table of coefficients that we're planning to put up on GitHub, we provide that same table in an Excel file within a sheet so that the sponsors can check it. So again, that's just something to have a think about. So key messages. We've had a great time using Vivli. If you are coming to something like Vivli, having had your own data on your own laptop, then you're going to find it quite challenging. If you're coming from a routine data world, it's great. The turnaround times are great. The data standardization is really good. We've had a great time. But there are challenges. And although the principles of data analysis are the same, the environment's not. So you have to think about tools for your pipeline. You have to think about how you're going to actually leverage the external and internal documentation in an efficient way. And as I mentioned now a couple of times, you want to plan for how you're going to share and export your data from the beginning of your analysis. So thank you very much for listening, and I will stop sharing.
Thank you so much, David, for your very informative talk. So I'm just going to remind people to put their questions for David in the chat. This is really a rare time to get time with one of our investigators who's so knowledgeable and has done quite a bit of work in the Vivli platform. So I have a couple questions for myself, and then if there's any questions for David, I also have a couple questions for you, but let me answer the ones from myself first, and then I'll ask questions for David. So in terms of questions that are coming from me, does it cost anything to use the platform in different environments? Yes. So those are all published on our website, but just to be more specific, in terms of the charging, we do charge after 1 year. The data use agreement runs for 1 year. And then for the standard environment, there is a nominal cost of $15 a day to use the environment. For the more premium environment, it does cost a bit more after the 1 year data use agreement is up. So that is the answer to that. Thank you. And in terms of the second question involves whether we would consider adopting any LLM software to expedite data harmonization processes. We've actually never done that, but if there's anyone who, and I think that may come down the line, I think certainly LLMs are powerful. There's also there's a tension between using LLMs to expedite the data harmonization process, which is really a wonderful idea, and the possibility that they are so powerful, and there's somewhat of a concern that they may be able to re-identify participants. So I think that's where we are, but certainly welcome a conversation with anyone who has that type of expertise. So I think a question has come in about, I don't know, David, if you can see this clinical trials toolkit in Pinnacle 21, how it differs. Maybe David, you want to take that question.
I'm afraid I am not familiar with Clinical Trials Toolkit.
Oh, I'm not.
I could tackle that.
Okay, great. Thanks.
Kausalya works with me. So Kausalya, the Clinical Trials Toolkit in Pinnacle The former is a SAS tool. The latter is a Pinnacle tool. They're entirely different pieces of software than Vivli. Vivli is a data sharing platform. The SAS Clinical Trials Toolkit is somewhat of a deprecated CDISC tool set, and Pinnacle 21 is the commercial tool that works with CDISC standards and is tied in with the FDA. So they're all entirely different tool sets. Vivli is a data sharing platform. That's what makes Vivli different. Sorry.
Thank you. Thank you, Jack, for answering that for us. So my question to you, David, is what next? What do you think is the next thing that you're going to embark on in your efforts?
Well, we've still got a few papers that we need to do for this grant, so a bit more of the diabetes stuff. But no, there's lots of things we're really interested in this space of kind of multimorbidity and representativeness. I think we'd like to dig into the adverse events and the serious adverse events a bit more. We're really interested in this kind of overlap between kind of real world participants and trial patients. So we've got a few different grant ideas, and also I sit, and if people are familiar with NICE, in one of our in the UK we have this cost effectiveness review process for drugs, and I'm on one of the panels, and quite often questions come up like, how do treatment effects wane over time? And often I'm sitting there and I'm thinking, gosh, we really could answer this with the IPD. So I'd love to get a program grant or something like that to really just kind of sort of be able to just, hit all the questions that really keep coming up again and again. So I'd love to do more of that. Another thing to say as well is I'm delighted to work with other people if they want to learn about our experience. And similarly, if we can find a way to do so within kind of privacy, I'm happy to share code, happy to kind of have things reused.
Well, thank you so much. That's a very generous offer, David. Wonderful. So I have a few other questions in terms of sharing with the sponsor. Is the research independent of the sponsor? Could you answer that, David? Are you working directly with sponsors on your research? Or are you independent? And how do you work with them?
So it's very much independent. I mean, I'm certainly not against kind of sort of collaborative working, but that's not the model here. And actually, it's quite an interesting question because sometimes the assumption from reviewers or other academics is that you've got a relationship with the drug companies and that's what led to the work. But no, I mean, you apply to the data, there's an independent steering committee, and the sponsors, they might have some comments on the data and so on, but really it's incredibly independent, so much so that it doesn't interfere with any of the work that I do with NICE.
Great, thank you so much. Yes, so we are an independent data sharing platform. They are not co-authors in any respects, but you do acknowledge them in your acknowledgements that you did receive the data from whoever it is, BI or Roche or whoever. For one single research proposal, if there are different people working, how is that handled? How many people can have access to the same research proposal? And how much does it cost for each single access? That's a good question. I mean, do you have multiple people on your team, David?
We do. We have, so it varies, people come and go. You have PhD students coming and going, you have collaborators and research associates. So at the moment, we've got about 5 people that can access Vivli, and 2 can, because we're on the standard cost, any 2 people can do it at one time. So we have a little WhatsApp group saying, get off please, because I need to get on. But other than that, it works quite well, although communication was an issue at first. I think you really need to think about who's going to do what and how you're going to communicate things, because it is a controlled environment.
So what David is describing, at any one time, like simultaneously 2 people can be in the environment, but we don't have a limit on how many. There could be 5 people on a team, but at any one single moment, only 2 people in the environment. So there's not a cost for the access for for the first year, it's free for researchers to put in proposals, but after the first year, there's a nominal cost of $15 a day. And the last question before we flip over to Jack, if anyone wants to combine data with Vivli from other repositories, we link to many, many repositories so that those data can be brought into Vivli. So I want to make sure we have enough time for Jack. And then at the end, if you have other questions for David or myself, we'll take them at the end of the presentation. So I'm moving it over to Jack. I know you have a really interesting presentation. So Jack. And then again, if you have any questions at the end, we'll also be able to take those. Thank you.
I should be sharing now. Can you see?
I think it's coming in.
Okay.
Great. Looks good.
Hey, I just want to say thank you. This is an amazing opportunity. Thanks to Rebecca for Vivli. This is a treasured resource, a data repository of this size and value. Thanks to Nicole for the CDISC presentation, because data standards are awesome. And thanks to David for his pragmatic experiences in using CDISC data within the Vivli platform. So what I'm going to do, I'm Jack Shostak, Operational Director of Biostats at DCRI. I'm going to take you through CDISC a little bit. Now, keep in mind that there's the SDTM and ADAM standard is like 1,000 pages of text to learn it. And it's a lot. And CDISC does teach classes on this that I'm not going to touch in 45 minutes. But the resources are there for you. All right, let's get moving. All right, so why are data standards important? They facilitate data use and efficiency. And like, if you know the data standard, then you can go from one study to the other fairly easily and find what you need. So data in the same location, data in the same format. And then, it helps you automate your analyses, use similar analytic tools, reuse code that you used before. And then what David's doing is allows you to pull data across a lot of studies for meta-analyses. Now, what does it look like? Something like this. So adverse event data in the SDTM is always going to kind of look the same. It doesn't matter what study you're looking at. You're going to have a subject identifier, right? You're going to have study identifier. You're always going to find the coded adverse event term in this field. It's always going to be that way. And the theory is you can take all these studies and stack them up like Legos, and you're good. So that's the hope. What is the SDTM? It is what David is using a lot. It is the study data tabulation model. And think of it this way really simply. It's your collected data, right? So it's the IPD, if you will. Individual patient data. It's the SDTM. Down here, go to cdisc.org/SDTM, and you can read all about it and learn about it. But there's two key documents you want: the tabulation model and the implementation guide. Again, it's about 250 pages or more, but good resource if you're trying to learn this on your own. Now, the SDTM is organized in what we call domains. And again, the SDTM was largely created by data managers in the industry. And so we think in terms of this: demographics, concomitant medications, vital signs, adverse events. So those that work on clinical trials, this is how the data is collected: by form, by domain. So the SDTM is organized by domain. And yeah, that's that. Now, There's a lot of standard domains, and you can create custom ones as well. But that's the substrate. I do want to recognize Atom as well. So the SDTM is the collected data. Atom is the analysis data standard, and it is built on top of the SDTM. And so, again, much like the SDTM, you can find all the documentation on Atom at cdisc.org. And again, there's two documents. There's the model document and the implementation guide. Again, it's hundreds of pages of text, which I'm going to try to summarize in 45 minutes. But this is, you've got your collected data, and then what statisticians like David and others do, which is transmogrify it, modify it, create value-added data. And that's what we put in Atom. Now, how does Atom relate to the SDTM? Fairly simply. Atom is built on top of the SDTM. So the SDTM is the studied data, it's the collected data. Atom is the analysis-friendly version. So from experience and from industry, we consider Atom to be the gold standard for analyses. So when things get submitted to the FDA or PMDA, it's off the Atom data sets that we produce our results. So, and Atom is designed in its very nature to be traceable back to the SDTM. And the idea is this, and it's hard to see, I know. So you have a table in your clinical study report, and it looks like that. And then you can trace it back to the Atom data set, which you can trace back through the metadata, which you can trace back to the Atom metadata, to the SDTM metadata, back to the annotated CRF. So in theory, with CDISC, I can have like a descriptive or inferential statistic here, and I can chase it all the way back to the raw data. Transparency of what we're doing is the key, and we call that traceability. Now I want to talk about the SDTM, which is what David talked about a lot too. So again, SDTM, it's your collected data, right? Very little derivation in there. It's mostly as collected. The Atom data is the modified analysis data. A couple critical things that go with the data is what we call define file. And that is basically an XML file packed with metadata that you can readily look at in a browser. And a thing we call reviewer's guide. And for the SDTM, we call it the study data reviewer's guide. It's a document. It orients you to the SDTM. Again, key concept of the domain here. So the SDTM is organized by domain. So demographics, adverse events, exposure, that kind of thing. And then within a data table, a domain, there are variables, columns, and they're standardized. And so again, here, you're going to have adverse event term, which is where you're always going to find the coded adverse event term across every study. Very helpful. And then this concept, and this is the beauty of CDISC too, this concept of metadata. Not only did CDISC bring standards to the table, but the real power is the metadata. It's the ability to have information about those data sets that we can leverage with computing scripts and software. All right, so where's the SDTM come from? All over the place. Historically, paper forms back in the day when we had paper data collection forms. Past 20 years, electronic data capture systems. and now and into the future EHR. So even though the source of where the SDTM comes from, it's still the same thing. It's collected data. It just depends on where you're getting it from, whether it's paper, electronic, or the EHR. And again, it's a collected data. It's what's submitted. Missing data are missing. You know, in the SDTM, again, there's no real, I want to say there's none. There's very limited derivation stuff going on there. That's on the atom side. And again, here, SDTM does not specify what data to collect. It's the study that tells you what you need to collect. SDTM just gives you a way to do it, a standard way to do it. And so here's what it looks like. Here's a sample data table. Not shocking to anybody that does research. It's a data table, right? It's got columns and rows. The variables, like if you were looking at it, they have column headers, tell you what they are. And then each row represents an observation or a record. Here, again, it's the collected data. So down here in the middle of this overlay, this would be like the eCRF, right? So you might have a collection form. In this case, it almost looks like it's paper-based because I can see writing on it. But this gets translated into the data table behind it. So we have this demographics table, which turns into a standard data table representation that you get to work with, and that is found in the Vivli environment. And so there's your study ID, there's your site ID, there's your subject ID, race is collected there, sex there, and so on and so forth. Remember, I said that the SDTM is a collection of this thing called domains. You can see a lot of them on the screen here. The SDTM has been growing for years. So over time, people add new domains. And you can add your own. It's not uncommon. You might have for your, like, these are all standard, like, things people run into running clinical trials. But obviously, every study's got different efficacy. So you're going to have, you know, you might have other domains not seen here that represent your efficacy endpoints. And again, go look, go read. If you're interested in the models, can't say it enough, go read about it. It's free. It's on the CDISC website. If you're confused by the SDTM, go download this stuff, read it in your spare time. It's a good read. Now, it's not just the data sets, and I want to be really clear about this. There's a thing when we do submissions called the define file. It is an XML file. The XML allows it to be machine readable. And so all the data sets that go in for a submission go with this define file. And it's got a lot of really good metadata in it. It's got data set metadata, it's got variable metadata, all this stuff packed into the define file. Really useful. It looks something like this. This is a screen grab of a browser. And like right at the start of the define file, you can see these are the data sets you get. And it tells you what they are and how they're structured. It even has hyperlinks on it to open the files if you have software that will open the files. And then there's helpful commentary. Hey, go look at the reviewer's guide. There's more detail on the demographics file in there. Might say what we did with screen failures, for example. And then you get into each individual data table and it tells you about all the variables in the data table. So what's the control terminology on the variable? How was this thing derived, right? And so mostly in the SDTM defined file, this column isn't super informative. I mean, it's nice it tells you where on the CRF page this thing is, but there's not a lot of derivation in the SDTM. On the atom side, there is. Don't you worry. So another thing that's really useful besides the define file with the SDTM are these annotated CRFs. And it's just we like pictures, we're humans. And so you can look at this form and tell where your data is in the database. And so, all right, here's my demographics form. I want to get the race variable. Oh, look at this. It got chopped up. It's in this thing called SupDM. That's a big hint, and we'll get to that in a minute. But you see where the birth date is, sex, ethnicity, race, the date of assessment, what visit is, all of that, just really quickly and visually, you can see where your data is. It's very helpful. Now, also in the define file, you get this control terminology, which is super helpful. It tells you the coding. So like if you've got like the treatment arm is in this arm and arm code variables, and you can see what's in there. Super helpful there as well. It's more control terminology. I'm gonna bounce through that. But again, it's in the define file. It actually shows you the mapping. So here in the data set, I might see 1-10, 1-20 placebo screen failure. And then in the define file, I can see how that actually translates into something a little more descriptive. Here's some examples of what these STTM data sets look like. And again, demographics, boom, annotated for the STTM, so I know where everything is. Thing I want to point at is this, like right here in the upper left-hand corner is telling me all this stuff's in the demographics domain, and there are annotation guidance to tell you how to read this. It is colorized so that the light blue tells me it's all in the demographics. If I had multiple domains on this page, I'd have multiple colors. But that translates into this data set. And let's look at the supplemental part, because this is a thing you should know when dealing with SCTM data. Not everything's parked in dm.xpt. A lot of the time, for a lot of these clinical studies, you're going to get information, I hate to use this word, but parked or shunted into a thing called supplemental qualifiers. And they all start with SUPP. And so there's your DM data set right there. That's the standard stuff, right? But the thing is, when you have supplemental qualifiers, they look like this. And the supplemental qualifier stuff in the SDTM is really a name value pair. So it is like, what's the name, what's the label, and what's the value? Where's QVal? QVal is the value. So it's not in the dm.xbt. You have to join these things together. So we'll get to that at the end of this. But the bottom, just know that a domain isn't necessarily just domain.xpt. It might be sup.dm.xpt. So sometimes you have to bring your data together. And I know David's going, oh yeah, tell me about it. But yes, that's what this is saying. The demographics domain is actually the DM data set plus these supplemental qualifiers. And sometimes, I'm going to tell you, oftentimes the data in the supplemental qualifiers isn't that important, but a lot of the time it's really important. So you can't ignore them. Identifying a subject, all right, so yeah, I'll mention this. So practically speaking, and there's a long story behind use subject ID and subject ID. Use subject ID is intended to be the subject identifier across the application. So if you had a patient who was in multiple studies for like an NDA, they would have the same use subject ID. And then within a particular study, they reference by subject ID. And I'm just going to tell you practically, next to nobody's using subject ID. Everybody's all the analysis, everything people does is pretty much on use subject ID. If you're confused by that. In the SDTM, there's also very useful information on treatment arm. And there are synonyms. So there's an arm code, which is the short name for what the arm is, what the person was treated. And then there's like an actual arm code and an actual arm, because sometimes people do not receive the treatment they were randomized to. So that's there for you in the demographics data set as well. Very useful if you're trying to do treatment comparisons, obviously. Also very useful in the demographics domain. There's a whole bunch of reference dates. And again, it's extended over time. Like if I were to go back here, the actual ARM stuff didn't really exist in the SDTM. We had it in the Atom side, and it kind of bled back into the SDTM as a useful thing. Similar thing has happened with reference dates in the SDTM demographics domain. So you have the reference start and end date, which is basically typically, and you go look at the implementation guide to get details, but the reference start date is typically data first randomization. And then reference end date is typically the last day on study. Now, there are other dates that are really useful, like when did they first get study treatment? When did they get last study treatment? That's these guys. When were they informed consent? And like when did they die if they died? And so there are very useful reference dates and key dates in the demographics domain. Yeah. And again, it has extended over time. These reference dates have built out in the demographics domain over time. There's a very useful exposure data set or exposure data sets. One is exposure as collected, then there's another one that's been distilled a little bit. It's just called the EX. But fundamentally, they're the same thing. It's what the patient was on when. So it looks like this, right? So you're going to have what they took. Here's the person, use subject ID, what they took, what the dose was, the units, the route, and all of these qualifiers on the intervention, and then the start and end dates. Very useful if you're getting into actual exposure data. It's a good thing to know that that's out there for you. Adverse events, beautiful, fantastic domain, because guess what? It's pretty standard across studies. And so again, you're going to see an annotation here. AE term is your friend. David had mentioned coding. Typically, this is going to be a MEDRA coded term. The start date, relativeness to the drug, severity, action taken, seriousness, end date, and so on and so forth. And there's a whole bunch of stuff that's not here on the screen. And so you can go look at the SDTM implementation guide. It will tell you in excruciating detail what all these fields are. But the beauty is it's standard. And I guarantee you, I can take adverse event data sets across a whole ton of studies and put them together. So that's fantastic. It typically looks like this, the AE data set. And it's, it's not really rocket science here. If you've seen adverse event data, then, and the little squiggly lines basically were stuck to the screen size. So imagine that just going off to the right indefinitely. But you can see all these standard variables. Again, my friend, adverse event term, this is the coded term. There is typically a verbatim term as well, which you're not seeing here. And if the data's been redacted and de-identified, you might not. Oh, wait, AE term's the verbatim, sorry. AE decode is the decode. And then beautifully body system, high level terms, system organ class. So a lot of things have been done for you to organize these adverse for the main study results when the study was submitted to a regulatory agency. Great stuff. Again, look down here at the dates. You'll see some partials. All the dates are ISO compliant date strings, so it's year, dash, month, dash, day, or as much as you know. But a really, really useful data set on adverse events for a study. Yeah, so AE term, verbatim, AED code is the MEDRA lookup. Okay. All right, so you want to bring your data together. The beauty is demographics is always one record per subject. So you can join it, merge it with any other data set in the SDTM trivially. That's all this slide is really showing you. It's showing you that these variables here are demographics variables, and they just got joined in with the adverse event data because maybe you wanted to look at adverse events by intervention. Seems like a reasonable thing to do. That's how you do it. You got to bring it together. The SDTM doesn't do this redundancy thing. When you get into ADAM, because of ICHE9, you do get some redundancy. So it's fairly common on the ADAM side to have the treatment variables already merged in with all your data. But on the SDTM side, you don't have that. So this is the individual patient data. You're going to have to do some data munging for sure. Disposition data set's really good. In the SCTM, it tells you where the patient was when. You know, were they randomized? Did they complete? When was their informed consent? So all these subject disposition events are going to be in there. They're coded with standard terminology, and there's dates associated with the event. So if you want to follow a patient through the study, this data set could be very useful to you. And it looks like that again. So there's a controlled terminology. Well, there's this term typically coming off the CRF. The D code is kind of SDTM terminology. It's a suffix for, this is the coded term. And then there's some categorization done on what type of event it is. But again, it's all standards based. It's all described in a lot of detail in the implementation guide. Lab data is fantastic. Been working with CDISC for about 25 years, and lab data is one of the first data they attacked. Why? Because labs all around the world were having to do custom lab exports for all of the various proprietary users of the data. And honestly, y'all, it's crazy. Like lab data is lab data. It's like name value pairs. What's the name of the lab? What's the value? But the beauty that CDISC brings to the table in these data sets is like things have been standardized. So you can come look at the lab data set and here's the lab test. And great, fantastic. And there's all kinds of qualifiers in here. And some, categorized as chemistry, hematology, urinalysis, that kind of thing. But this is where the LB domain shows its power, because here's the original result, and anybody that's dealt with lab data that's not been harmonized, there's pain involved. So there's an original result in the lab data as it was collected, and then there's a standardized result as well. Now it's not on this screen, but there it is. So there's the standardized result, and I'm going to do the analysis. This is what I'm using, the standardized result. And so basically, a lot of times with lab data, if you've got local labs, the labs are coming in with different units and things like that. The standardized results always standardized on a common unit. So bilirubin's always going to be in micromoles per liter, period. And so this is a huge gift from CDISC at the SDTM level. Remember, I said there's not a lot of derivation here, but the lab data has been harmonized for you. Much like the adverse event data has been coded for you, the lab data has been largely harmonized for you. So very, very helpful. And then a whole bunch more qualifiers in here as well. Again, this was one of the first things CDISC tackled like 25 years ago, and thank God they did. And so you see it represented here in the lab domain. And basically all the lab vendors in the world speak in this language now, which is awesome. It's fantastic. All right, we're going to talk a minute about ADAM. And I'll try to make this brief. So what is ADAM data? So we talked about SDTM. It's the collected data substrate. ADAM is the analysis data. So the idea, fairly early on with the SDTM, we're like, this isn't great for analyses. We need to do stuff to it. We need to torture the data. So we did. And we wrote this thing called ADAM. And it's a standard model that supports consistent and effective use structure of analysis data sets. The hope is it helps be clear and about what we did to the data to torture it for analyses. And it's designed to help a statistical reviewer at a regulatory agency trace back from our analysis data sets to the collected data. And the hope being is it allows for efficient generation, replication, and review of results. Make the reviewer's job easier, be more transparent, show them what you did, and let them pick it apart. In these Atom data sets, they're a hodgepodge. It contains SDTM content as is. It contains derived things from the SDTM content, and it's not domain centric. So a single Atom data set, especially if you're doing like a survival analysis or time to event analysis, a single Atom data set can contain data from like 10 different domains from the SDTM easily. There is a lot of work put into these Atom data sets. Much like the SDTM has a reviewer's guide called the Study Data Reviewer's Guide, the Atom data sets have an analysis data reviewer's guide. And again, just looking at data tables and metadata can be painful, so you want to document so you understand the data. The Analysis Data Reviewer's Guide is not yet required by the FDA, but it is required by the PMDA in Japan, so that's kind of fun. You're generally going to have it with a submission. It's always going to exist, and I won't go into why right now. But it provides really good information about the analyses that was done in those analysis data sets. And yeah, there's some stuff on conformance findings too, but don't worry about that right now. There's three basic Atom structures. There's a subject level analysis data set. It's kind of like the demographics data set in the SDTM. It's one record per subject, has a whole bunch of really useful variables in it, and it's critical. Then there's this thing called the basic data structure. Think repeated measures type analyses, one or more records per subject per parameter per time point. So it's just this big vertical stretched out monster. And then we have this thing called the occurrence data structure, which is really, it's for adverse events, medical histories, think occurrence type structure data, which is often stored in hierarchically compartmentalized data. Analysis data set subject level. You can see the screen contains critically important variables. It's always one record per subject, which is beautiful because it allows you to take ADSL and join it against any other CDISC data set in a submission. It's the source for a lot of critical variables as well. And it allows you, right out of the box, it allows you to do a bunch of things. Like you can do like most of the table 1 baseline comparability stuff that you would have in a clinical trial, you could get right out of ADSL. It's fairly easy. Why did Atom create this basic data structure concept? Trying to standardize. It's a really like, and I think David alluded to this too. The SDTM is more standard, to be honest. There's less variability. The SDTM is really put this data in this compartment, right? In Atom, it's more of what do I need to do to my data to get it analysis ready? And what I mean by analysis ready, I mean readily consumed by an R function or a SAS procedure and ability to churn out scientific data from it. So because it's more the goals of it's not stick this data in this bucket, it's how can I organize my data to make the analysis more efficient? That's what this is. And the basic data structure is an attempt at a singular way to do that. So that's what we did. All right, this is the fun part, and this is where I hope we get some good exchange on in the chat that's coming up. Understanding your CDISC data in general. So let's say you go to Vivli and it's a bunch of CDISC data sets and you don't know CDISC at all. It's a challenge. my advice to you is try to learn a little bit about CDISC. Go to cdisc.org, read up about the SDTM, read up about Atom. If you're doing this a lot, take a class, learn about it. Also, if at all possible, get the reviewers guides, get the study data reviewers guide and the analysis data reviewers guide, if at all possible, and then the annotated CRF, because that is going to get, it's really interesting listening to David's talk because he was talking about leveraging ct.gov data, which is fabulous and brilliant. But I'm telling you, if you have these artifacts, they're going to help too. And this gives you a window into what the supplier of the data did for the primary analyses. So really useful. Try to get that stuff if you can. So read up on the models, try to get the supporting artifacts that went with the original submission if you can get your hands on. This to this day, this is a question that reviewers at the FDA have about CDISC. Where's my safety and efficacy endpoint in the sea of data sets? I gotta be honest, it's not standard necessarily. You gotta understand the study. So, like, for example, if your primary endpoint is, I don't know, maybe you're working on a pain study and there's a pain questionnaire, you need to know that that pain questionnaire is where the key efficacy data is stored. Then you need to know that it's a questionnaire, so it's stored in the QS domain in the SDTM. And then you got to go find out what they did on the Atom side if you've got the Atom data sets too. So it's not, you can't just know. There's no standard CDISC standard data set that says my primary endpoint is here. That doesn't exist. And so it can be a little bit frustrating, and I get that. But you kind of need to know the study to know where to look in the SDTM and the Atom data sets. Now, this is a bias because I helped build the Atom standard. But go, if you've got the Atom data sets, go look there because there's going to be a lot of analytical work that has already been done off of the individual patient data or the SDTM data to get to those analytic endpoints. And for the folks on the call, I hope there's some people nodding their heads. There's a lot of work that gets done off of the individual patient data to get to an analysis ready data set. So if I'm a researcher and I can reuse that, I'm going to do it. It can save me tens, if not hundreds of hours. So highly recommend you look at the Atom data if you can get your hands on it. And some of these things can be wildly complex. They're on the Atom side, it allows you to have hierarchical analysis data sets, which goes to show you how complicated this can be sometimes. So again, if you can get the analysis data reviewers guide, look at that because there's going to be something in there in text, in words that you can read that says, go look at this data set. This is where I parked my primary endpoint that is referenced in my SAP in the protocol. Save you tons of time. And then also, if you got the Atom define file, go look at that because all the gory derivations and algorithms that are done to support the primary manuscript that's for the FDA or PMDA, it's in that define file. It's defined for you. So you don't have to root around in some arcane R or SAS program to figure out what somebody did. You can just see it in a little window in a browser. How do I merge my data? You gotta have a little bit of data sciencey skills to like and get anywhere near what David's doing. The data is standardized, but you gotta work with it. You gotta munge the data. You gotta play with the data. So, and then there's this challenge here on the SDTM side where data split a lot of the time. So you'll have a domain like ae.xpt, and then you'll have a sup ae.xpt. And so you got to join these things together. I guarantee you David's got one of these. They're out there on the web. It's public. You can get the stuff. But go find a program that just pulls these things together for you easily. Pretty much every company has them. You're going to want one if you're working with the SDTM data. You're not going to want to leave home without it. So get a program that does this for you. Tons of them out there on the web. They exist in R and SAS. And keep in mind too that, and there was a slide on this, SDTM DM domain and the Atom ADSL dataset are one record per U-Sub ID, always. And so they were designed to be joined and merged with all the other data sets as needed, which is also why the demographics domain has a bunch of critical date variables in it. And the ADSL data set can be sometimes overloaded with a ton of really helpful variables. What a data challenge. What if I go into Vivli and I only have the SDTM data and I do have an annotated SDTM CRF? That's good. But that means you're working entirely with the IPD, individual patient data, or raw data, as I like to call it. You're going to have to rederive things that may have already been done for it. So this can get a little bit challenging, especially if the data has been anonymized, because let's say you're trying to replicate the sponsor's original results, and there was some stuff that took the edges off the data, and you can't quite get back to it. So, knowing how they did it is key, because if I'm trying to rederive things and I'm already battling through the anonymization resolution issue on my data, and I don't know exactly how the original authors derived their result, it can get really hard. So again, this is a plug for try to look at the analysis data sets as you can. Try to look at the reviewers' guides if you can. If all of you guys, the SDTM and the annotated CRF, you're gonna be okay. It's just gonna be a little bit harder. And David mentioned this one too, and I think it's really important. The data on the Vivli platform has been anonymized. And so what does that mean? That means, like I described it as taking the edges off the data. But the data is not necessarily what the original authors had. Well, most definitely isn't. So that can be a challenge. Also, if you are lucky enough to get these artifacts, the defined file, the reviewer's guide, it's very likely that they were created on the fully identified data. So you may look in the metadata and it's not fully descriptive of what you're seeing in the anonymized data. My comment there to you is this bit right here. Definitely try to find a document describing how the data was cleansed. Find a document on how it was de-identified and anonymized. So when we're producing those things to store on the NIH data repositories, we always have a document that describes what we did to anonymize and de-identify the data. So that will help you understand how the resolution on the data may have changed. There may be things you can't replicate anymore. Some of the identifying data may have just been redacted entirely, so you can't get back to that point, but it'd be good to know that. And yeah, so just understand that data anonymization might prevent you from fully replicating original study results. It's just the issue. And that's all I really have. I'm happy to take questions. Yeah.
Thank you so much. Let me share my screen and
Yeah, I'll
go to
stop.
questions. So let me have, well, thank you so much. I thought the common data challenges part was extremely helpful and not some of the things that I actually had thought about. So that was super helpful, actually. I'm going to have David kick us off. And let me ask David about a couple things first, and then I'm going to ask Jack. Some of the things I thought would be helpful for you, David, to elucidate on is what you would do differently now that you've heard Jack talk, if anything.
So first of all, I think I would have learned what I learned a lot more quickly and less painfully. So a great shortcut. I mean, a simple example of that is, understanding about the standardized lab variables. And even though we didn't want to use the SI value for HbA1c because it's more common to use percentage. But actually, we would have been better taking the standardized one and then changing back to percentage. So that would have, even though it's 2 steps, it's more standardized. So we would have done that. I would have funded for our PhD students to go and spend a bit of time training in SDTM. And in fact, I've got a PhD student starting in October and they've got a bit of a budget, so we'll spend some time on that. I would drop some of the character variables sooner, I think, because they're already in the numeric ones, so just simplify the data. I would love to get my hands on this define file. I've never seen one. It's like a zebra or a rare dragon or something, but the idea of rooting around CRFs to find out what does number 3 mean inside the smoking status variable, if there'd been a machine readable thing, that would have been fabulous. So yeah, no, we would do lots differently, and I think we would have done it in about half the time if we'd heard this talk first.
Wow. Okay. So I'm so glad we did this 7 years in.
It's never too late. It's
Yes.
never too late. Yeah.
Yeah. So thank you. I think you're just inviting yourselves back for next year. So start booking your calendars. Yes, Jack, any insights from David's talk before we keep?
It's just really like the one thing he was talking about leveraging CT.gov and the way he did it is it's super cool, but I'm like, man, if you had the define files, that'd help too. So the initiative to go reach out to that resource is brilliant. But like just something to keep in mind, like, and again, CDISC really educated the industry on metadata and how you can have data about your data and leverage it in consistent ways. So to just have the data tables is kind of, you want that metadata. And then also the reviewers guides. And so like, I would just push for that. I would push for that because I think it'll save David and folks like him a lot of time. And they exist. Anybody who has CDISC data sets, well, 99% of the time, unless it's a failed study. If it's a failed study, it's very possible that the pharma didn't complete crossing the T's and dotting the I's. I've seen this. But if it's a completed study that got submitted, I guarantee you there's a defined file and I guarantee you there's reviewers guides. They have to submit them. And so I would grab those things. They exist and they will save you time.
Rebecca, can I come in there? Because I think it would be lovely if the sponsors were even able to make the define files publicly available. Because that would be great. If you're looking on Vivli, you're looking at trial, if you can download that define file, you've got a really clear idea about whether or not the trial has any chance of helping you answer the questions you're interested in.
Yeah, very good perspective.
I will just one tiny caveat to that. The defined files are metadata. So it's data about the data. Largely speaking, there shouldn't be anything identifying in there. However, there's nothing to say that somebody can't put in there, if subject equals Bob Smith, then hard code some value. So I have had some sponsors say, oh, we can't give you a defined file because that's identifying, which I immediately turn around and say, it really shouldn't be. So you might run into a little resistance there, but I don't buy it. And look, honestly, y'all, let's be very clear. Thanks to Juan He and others doing a lot of stuff with large language models, anonymization and de-identification won't be with us for very long. I'm just saying. So, yeah, if somebody says they can't give you a defined file because it's got identifying information in it, shame on them. It probably shouldn't. And 99.99% of the time it doesn't.
Okay, thanks. So let's go through the questions. Now that clinical research is taking a more global approach in the wake of the COVID-19 pandemic, how are you planning to deal with selective availability bias to enhance generalizability when the data available on Vivli is not uniform across regions, populations, perhaps due to various factors such as regional representation, underrepresentation in clinical research, eligibility criteria, or perhaps selective reporting? Either of you could answer this question.
I guess I would say that the role, so Vivli is about secondary data analysis. So I think the role of Vivli and people using Vivli is to delineate that problem. Because we all say that trials are unrepresentative, and okay, that's fine, but how unrepresentative are they? For whom? You know, we need to, there are trade-offs here. So basically I think that Vivli has the role of better describing the problem.
Hey, well, thank you. I can take the next one, which is how can we help AROs or CROs who work on behalf of sponsors, but not entirely on data sharing? And I think the question really is that these CROs, and I've worked with a CRO before, is they tend to hold the data they tend to hold the data on behalf of sponsors. So I think Vivli should be working more closely with CROs as they hold the data and for data sharing purposes. And then we're happy to connect with any one of you if you'd like to work with us directly. There is someone who suggested that the defined file would only be for submitted data, and others such as phase 4 and terminated studies would probably not have the compiled data defined file. And I think that's certainly true. I do think if sponsors would be willing to move forward with making those more publicly available, I think that would be certainly very welcome and make the request process much more smooth for requesters like you. And I think we could certainly work with that. There's a question on probably the interpretation or definition of anonymized data. We get asked that quite often. And many researchers ask the definition of anonymization versus de-identification. I guess the easiest, and does any one of you want to take a stab at that? Yes, Jack.
The huge problem, a huge problem. So anonymized in my mind is the common rule definition, which says you can't get back to the original subject from the data that you have.
Yes.
De-identified in my mind is the HIPAA definition of de-identification. Now, that said, everybody does it differently. There are different, like David will tell you, he's like, there's different, like everybody's anonymizing and de-identifying differently. Some folks use like data jittering for dates, and I don't do that. I don't believe in jittering. I think it's silly. So everybody's using different techniques for anonymization and de-identification. It's a huge problem in the academic space, to be honest, that there's not a collective definition. I will say that the common rule in the US and the HIPAA definitions around it are something to sink your teeth into. That is the closest I could get to a common definition. But the practicality of it is, it's kind of implemented all over the place in different ways.
Yes, absolutely. And even among probably what you've seen, David, is you might get 3 data sets, or you've asked for much more than 3 data sets, and they're anonymized in different ways. And how do you manage that?
So part of the management of that would be, so for example, we've done a lot of work on this frailty index developed by Ken Rockwood in Canada. And the concept there is that you can measure frailty if you have lots of different components. If you've got 40 to 60 components, and it doesn't matter exactly which 40 to 60 components you have, as long as you've got 40 to 60. So it's a little bit like that. Or, so for example, for defining comorbidities, if we got the medical history at baseline, great, we use that. If not, we use concomitant medication. If we had both, we used both. So as a routine data researcher, this is life. You know, like someone comes to the clinic and they get their blood measured, and someone doesn't and they don't, you know. So this is our bread and butter. So yeah, I think you've just got to be practical about it, really. One point I would make as well is in the UK, we don't really talk about anonymisation so much. We more talk about pseudonymisation, and we use safe havens. So it's a slightly different framework.
Yeah, there's that too, right? The global like GDPR versus HIPAA and things
Yes,
like that. So it's you have my sympathies, Vivli. I mean, it's a hard space.
it is a very difficult space. Absolutely. Okay. So I would invite Nicole back on so we can maybe just wrap up. And just ask you to see if you have any closing remarks. So we can start with you, Jack. Do you have any, thoughts on any closing remarks or any reflections after today on where this might go in the next few years? And I'll
Yeah,
ask the
I
same for
mean,
Jack and Nicole.
um, I would say like a few years ago, I was like, Vivli is awesome. Vivli is kind of the big data sharing platform on the planet, but there's tons of them. So there are a lot of data sharing silos. Like a lot of pharma spin them up, a lot of academic institutions spin them up. They're typically not standards based. They might be like Vivli, where some is and some isn't, and it makes it hard. Also, Juan, he had mentioned LLMs, and it's like I think for a long time we've tried to structure data to make it easier for us to use. And part of me now looks at it and is like, we actually didn't help the machines. Like I would argue an LLM would much rather read a clinic note than get a data table from me that says MI at the top and has ones and zeros in it. And so it's kind of a weird time. There's a lot of promise in large language models and what they can do to help bring order to the chaos in the data source that we have. So I look forward to that. I also think there's going to be a huge challenge around anonymization, de-identification. Our digital footprint is just too big. And I think it just realistically, I think the future of that is like privacy is not great. So
Yeah.
sorry.
Thank you, Jack. David?
So I think that it would be good to see academics depositing their data a bit more as well. I think it's definitely easier to get industry funded data than it is academic. I think editors and journals probably have a bit of a role there as well about data sharing. So I'd like to see more of that. I think I'm probably a wee bit more optimistic about standards than Jack. Maybe he's battle scarred from years of it. I think that LLMs are great, but I think that structured data is pretty important as well, especially in a kind of space where you want traceability. But although I mean LLMs are great, especially for data extraction and things. So I think it'll be a really interesting space.
Absolutely. Thank you both. My perspective is I agree with you in terms of the academics. I think academics largely use our platform. I mean, 90% of the proposals are coming in from academics. We certainly don't see as many contributions from them, but we do see that number growing. I'd say probably about 10% of the contributions on Vivli are from academics, and we do see that the future is bright, and hopefully that they will start contributing and using the CDISC standards. That is our hope. So thank you so much. I think industry is really, contributing the vast majority of trials right now on Vivli. So I do think the future is bright. We are growing dramatically in terms of the number of requests. And we're up 40% just this year. And so we're delighted to see that, of course. Nicole, your parting thoughts?
Definitely just a huge thank you to you, Rebecca, and Julie, and just Vivli in general. I mean, we've had a partnership for many years, and thank you for the support of encouraging that the data be standardized so that it can be shared easier and you can look across the data sets. And Jack and David gave all the great, more detailed information, but if anyone is interested in learning more, definitely visit the website, reach out to myself or Jack. with questions as you dig in and start using them on your own. And we're happy to be a useful resource as much as we can, but we're glad to continue to grow our impact to the academic community. And I'm glad that we had this opportunity today.
Absolutely. Well, thanks all of you, Jack, David, and Nicole, for being with us today. This was really a tremendous opportunity to bring everyone together and really the thought leaders. So with that, we close out the webinar. You'll receive the slides and recording. But again, thank you so much for joining us today. So have a good day and take care. Thanks everyone for participating.
Thank you.
Thank you.
Thank you.